Good morning, everybody, and thanks very much to the organizers to getting this conference together. It's a privilege to be here. So we hope the technology will work. I'm Katharina Fabricius. I grew up in Germany, and in 1986 I heard a talk about coral starfish when I was a student and decided that's what I want to do in my life. And it was actually from Ames, and it was Russell Reichelt, many of you know, giving a guest lecture on coral thorns, and I thought Ames is the best place in the world to study coral reefs. I really want to solve the coral thorns issue. In 1988, came to Ames in 1988. Been in many places around the world since then, but I've had the privilege to work on the Great Barrier Reef and coral reefs all around the world since then. At present, we are very strongly involved in the Reef Restoration and Adaptation program. And obviously, reef restoration cannot happen without ecological underpinning. So we've been having the chance to put together a big program called, we dub it EcoRRAP, to provide the ecological underpinning to do the interventions that the reef need. Before I start, I would like to acknowledge the traditional owners, the Aboriginal and Torres Strait Islanders of the land where we are working and meeting today. I acknowledge the elders past, present and emerging, and I also acknowledge the present elders having given us the consent to work on their sea country. So for those of you who are not familiar with the RRAP program, Reef Restoration and Adaptation program consists of a whole number of programs, sub-programs that develop interventions and a number of cross-cutting sub-programs. What I'm talking about is the Ecological Intelligence for Reef Restoration. And as I said, we're dubbing it EcoRRAP. The objectives of EcoRRAP are to provide the underpinning. There's so many knowledge gaps. We cannot understand all of it. We cannot support reef restoration if we don't know what species are living where. We've got very little information on vital rates of corals, for example. We've got very little information on what are the limiting factors in reef recovery. So our purpose was to really develop a 10-year program to look at what is happening in the reef and how can we support reef restoration through ecological knowledge. That data is critical for the ecological models that are being developed in parallel, for the decision support, and also for the development of deployment guidance that is happening as we speak. So the Great Barrier Reef is obviously a hugely diverse ecosystem. It is as big as Italy, with 4 degrees temperature gradient almost from the north to the south, which means a projected 2.7 degrees warming is well accommodated in the reef if we are smart about it. But different regions are warming at different rates. The south, that's a very old and outdated graph. But if you look at the blue bars, you can see the southern areas being warming 3.5 times faster than the north. And so we've got a huge range of habitats, we've got a huge range of environmental conditions, and each reef region is different. So we've got to cater for different environmental conditions in order to decide what to do. For the EcoRRAP, we've set up 12 reference reefs up and down the Great Barrier Reef, from the Torres Straits up in the north all the way down to the south. The brown reef circles demarcate the regions in turbid water environments, and the pinkish ones in clear water environments. So you're really trying to cover the whole latitudinal gradient as well as in inshore to offshore environments for the GBR to set up these reference sites. And that's how it looks. In many cases, we also needed within reef information. So each reef has got sets in the sheltered and in a wave exposed area, in the shallow and in the deep. And so it's a very complex set, a nested design of reef plots and sites up and down the Great Barrier Reef. So we've got 88 sites, 350 plots to do the work. And those plots are really covering a very wide range of reef habitats, from algae-dominated areas to very hard coral-dominated areas, high silt environments, and clear water environments. What we're doing on those reefs is we are studying community dynamics, community vital rates of corals. We are looking at how the early life stages on the coral reefs are faring, and we're looking at cryptic species. It's not me. But we are not only looking at corals, we're also looking at species interactions such as macroalgae and the role of fish in determining reef recovery rates. So there's a lot of biological, physiological, and environmental studies going on at these sites. We'll see a number of these results throughout the next 3 days, and I'm just providing a quick overview of what EcoRRAP is all about. So Renata, who's here, has been key in developing photogrammetry tools to quantify things like structural complexity, which are a key predictor for habitat quality on reefs. If a reef gets disturbed, it typically gets flattened and is not as good a habitat for fish and reef-associated organisms. We're looking at vital rates. You'd think we know how fast corals are growing, but you would be surprised how few data sets are actually available from in situ in the field in relation to the environmental drivers. We're looking at species prevalence. Where do certain species do the best? If we're deploying corals, where should they go? Which species should we choose if we've got a certain site to deal with? So there's a huge amount of spatial ecology happening, and a lot of studies by PhD students, postdocs, and scientists. Relevant is obviously we need the tools to deal with this vast amount, really terabytes of data from those photogrammetry plots. So AI is being developed to extract segmentation for colonies, identification of species, to track colonies from one year to the next, and so on, to get the vital rates and to really get a better understanding of what the dynamics are on those reefs in different places. Those tools are also became useful now in the response to this 5th mass bleaching event we're experiencing on the Great Barrier Reef as we speak. So today you will have seen the 4th global mass bleaching event has been called upon by NOAA, and the Great Barrier Reef has experienced widespread bleaching up and down the Great Barrier Reef. On the left is a plot put together by Jess Roth from CSIRO, which shows the degree heating weeks. And literally, over 60% of the reef has seen very high or extreme levels of heat stress. And the results of the aerial surveys that Ames and Kubumba have conducted will be released tomorrow. So Torres Strait and the far northern Great Barrier Reef have fared, fortunately, a little bit better. Very few reefs have bleached up there, but the northern, center, and south has been very severely hit yet again. And that's the 5th mass bleaching event since 2016. It's not surprising the reef bleached. This year has seen 1.3 degrees above pre-industrial temperature ranges, and because the temperatures are rising so rapidly, we're going to see probably annual bleaching events as of well, initially they said 2040, but it looks like it may be happening much earlier now. So Renata and team have done the photogrammetry plots. Here's Heron Island in the shallow water, and you see the diversity of colony responses. Some colonies are badly bleached, others are faring better, and that's exactly the information we need to extract from the reefs in the wild populations to understand and to help the reefs recover and to predict and to accelerate the adaptation to these warming temperatures. That's a brief summary from in-motor surveys put together by Neil Cantin only yesterday. About over 90% of reefs had more than 30% bleaching in the northern. That does not include the far northern reefs, but 66% of reefs showed mortality. So it's really yet another really sad bleaching event. It's really happening, and that's why RRAP is becoming so urgent in order to understand how we can help the reef to recover. Obviously, relevance for reef restoration. We are in the process of developing tools to better quantify bleaching status, not just by in-water surveys, but also by photographic data extraction, quantifying how much a coral has bleached. We do need to understand the effects of bleaching on coral vital rates by having surveyed which corals in our plots have bleached. We go back next year to see how they fared. Has it compromised their growth rates, and, or have they survived and also come to the bleaching? Along the same plots, we are looking at fish communities, we're looking at macroalgae communities, so there's a lot of work happening here. Again, the AI tools developed to quantify fish densities. We are looking at the role of macroalgae as habitat for fish in many places. When the coral is missing, the macroalgae become the main structural element on the reefs. We are looking at the contribution of fish to reef health in terms of herbivory, corallivory, and we are looking at the role of macroalgae in diminishing the capacity of reefs of young corals to settle. Similarly, Doropoulos and others are doing a lot of work on coral recruitment, connectivity. We've been looking at juvenile densities, and I'm just showing you a few results here, which I only put together yesterday, so they'll be a little bit rough, but obviously, again, hugely important. We are moving corals, we are moving coral slicks, so understanding connectivity, natural connectivity between reefs, the drivers of coral recruitment, the environmental limitations in reef recovery are key sets of information for EcoRRAP. These are the data. They're much more complex models underneath, but I decided to go for blue and red bar graphs. Overall, the densities of coral recruits on reefs are still pretty high. So we are averaging over 90 recruits per square meter, and that's up and down the GBR. But there are up to 10-fold differences between reefs. Like Keppel Islands were the worst, and 10-fold higher densities of recruitment in places further offshore. In particular, Lady Musgrave and Lizard Island have been record numbers of coral recruits in the 2 years where we had the settlement tiles out. Remarkably, to go back, the red bars are the reefs in the turbid reefs, and the blue ones are clear water reefs, and you can start seeing the fundamental effects of water quality, which I'll show you in the next few slides, in determining coral recruitment. Coral juvenile densities, these are all corals less than 5 centimeters in size, remarkably similar patterns to the coral recruits that are arriving on the reef. So we really think we are seeing some recruitment limitation in the reefs in terms of the arrival of larvae and not later onset of survival issues. Again, the Keppel Islands had the lowest density of coral recruits. In the literature, it's commonly assumed that 6.2 recruits per square meters would sort of be the lower limit of reef recovery. Keppel's are on 8. We still need to refine those numbers, and Manu is working on those things. But at this stage, the red line here suggests where the coral recruitment may become limiting. In most places, coral recruitment is way above what the reefs would need to bounce back. But again, the 3 turbid regions have much lower juvenile densities than the clear water regions. And if you look at the ratio of clear water to turbid reefs, and again, this is a really simplistic display, but there are analysis behind it that support it, it shows that the ratio of recruits on the tiles is 3:1 between clear and turbid reefs, and the ratio of juveniles is 2.5:1 between clear and turbid reefs. So water quality really matters when it comes to coral recruitment and reef recovery through that mechanism. There's obviously coral growth and mortality as additional factors to consider. But interesting, the ratio of recruits to juveniles is about 5:1, both in clear water and turbid water reefs, which again shows that it's not later survival once the corals have settled, but it's the early settlement and post-settlement numbers that seem to be the critical bottlenecks for those reefs in the turbid environments. We also use the tiles to look at carbonate accretion. Basically, in this case, it's a fairly simplistic dry weight an assessment of how much these tiles had accreted in 2 years' time. Again, the Keppel Islands have been faring the worst, and the rates of carbonate accretion was much higher in the south than in the north. So there are some differences again in water quality. A ratio is about 1.5:1, with much higher accretion in areas of lower areas of sedimentation, and in particular the more than twofold differences in the lower levels of sediment weight, and that really makes a difference whether carbonates are accreting independently of what corals are doing. I mean, reefs are accreting through coral growth, but when the corals are missing, they still are being eroded through wave erosion and so on. So we need to know how much is actually happening on those 80% of reef area that are not covered by corals, and what's going on here. So again, a water quality issue here in terms of reef accretion, and again, turbid Keppel Islands are unfortunately struggling a bit in keeping up the maintenance. So again, I mean, the relevance for RRAP is pretty obvious. I'm just throwing it out to re-emphasize it. What does it all mean? The reefs are facing incredible pressure. Now we've heard a good summary for Uncle Bob, sort of pointing out the critical time we're in. We're really in a critical decade after hundreds and thousands of years of relative stability. We're changing the environment in an unprecedented rate. The reef disturbances are becoming more frequent. And so here, the little schematic I put down here is much better done by the modelers, but what it tends to show is we did have natural disturbances in the past, and there are capacities of the reefs to recover until now. If the frequencies of disturbances are increasing as it does now, through bleaching, through more intense cyclones, through more intense floods that bring more sediments into the water, because the warmer air holds more moisture, therefore rainfall intensity becomes more extreme. We are facing a time where reefs are becoming more often disturbed. So RRAP stuff will hopefully be able to, we can't prevent the natural disturbances, but we hopefully can help the reef to adapt to the warming temperatures a little bit, so we are not facing a wipeout, but some sort of dynamics of some semblance of coral reefs as we've known it in the past. So in conclusion, I believe at present all the data we've got are that most coral reefs are still quite resilient, but there are exceptions. The Keppels are one example where the reefs seem to be struggling a lot. Keppels have got high sedimentation, got the Fitzroy River to contend with. But many other places show very high recruitment, high carbonate accretion rates, and coral reefs are naturally dynamic. They are able to recover from natural disturbances. So the main problem now is the frequency of disturbance, but the dynamics are such that disturbances are fast, but then it takes years for reefs to recover, probably 10 to 30 years. So assembling those life history data takes time. EcoRRAP started 3.5 years ago, and we've achieved an enormous amount of time, but it has been designed as a 10 years program. So we are only just starting getting those information of vital rates, how fast the coral's going, or the reproduction rates, and so on. So it's early days in terms of showing you results, but there will be about 10 talks for the next 3 days showing you some of those highlights of the results that I've summarized here. We can now answer more questions about heat tolerance, about we're working on issues like connectivity, But nevertheless, it is a critical decade. Without climate mitigation action, the reefs will have very little hope. But the water quality links I've shown you here also shows very strongly that traditional reef management action is still immensely important, and probably more important than ever, because we know it works. We know water quality leads to better reef resilience. We know fish are really important in allowing reefs to recover. So we don't replace the traditional management with some fancy respiration. We need more of those things to really support the reef into the future. Yeah, and that's a big thank you for you to listen. The work could not have been done without those 45 people who are the core EcoRRAP members. I highlight in bold the people who may be talking in this conference. And I would like to really thank the RRAP managing entity, the GBIF, the conference organizers, and all the partners who have been supporting this RRAP program in this journey for the first 3.5 years. Thank you.
All right, we have about 5 minutes for questions, and we'll be doing a dual approach. So we have a Slido assistant in the corner here. Yvette will be assisting throughout the day with Slido online. And we ask so that the online audience can hear that you line up for the microphones if you're here in person and would like to ask a question. Thank you. She can repeat your question if that's easy.
You have talked a lot about the relationship with water quality and the sediment and turbid waters, but what about the nutrients? Is any indication as well as part of your research that is showing that relationship with high nutrient levels?
Yes, so there's a lot of research going on the effects of water quality on the Great Barrier Reef, and I've been involved in that, as you know. Water quality is difficult to tease apart which factor in particular is affecting things, because in many cases sedimentation and nutrients are highly confounded. So yes, we know that dissolved inorganic nutrients make algae grow faster, sediments affect corals more. So in many cases there are different pathways towards different effects. High nutrient runoff from cane farming in particular is of key concern for us. High sediment runoff from, in particular, grazing areas is of high concern. So there are regional differences, and in this sweeping talk I can't really go into details, but nutrients and sediments are both a key importance for the coral reefs. That's work in progress, and because we've got these photogrammetry plots that give us species compositions, we've also got the species composition in juvenile densities on the tiles. On the early recruits, it's impossible to tell them to do anything greater than families. But yes, these relationships, we're just calling for postdoc now to look at those relationships in detail, because it's a beautiful data set.
We'll just do one last question in person and then one online if there are any online. I'll repeat the question on the microphone. What? Sorry? Yep.
Is there any way to tell the difference from the EcoRRAP data on, say, for example, the site that's showing poor recovery, whether it's actual larvae in the water that larval supply is low, or whether the larvae are there, but the substrate conditions in the river just poor settlement due to the, if you can't actually measure the quantity of larvae in the water, all you're looking at is the settlement or the recovery. How can you actually work out what the recovery is if it's supply or sending conditions?
So to repeat the question, is it possible to use EcoRRAP data to tell whether recruitment is limited by larval supply in the water column or whether it's limited by available settlement area and conditions?
Thanks, really good question. And probably a third of the EcoRRAP program looks at the early life histories, and that's led by Chris Doropoulos. There are whole projects looking at connectivity, larval supply issues, and trying to tease that apart. But we haven't got the answers yet in this particular data set. So, but it's a very important question, and we are working on that. Thanks.
Do we have any questions online? Okay, thank you very much for opening this session, Katarina.